Last presentation for today, so thank you for staying. We're team one, we're MEViD, the Multichannel Electrochemical Viral Diagnostic. Imagine your child wakes up feeling sick. Over the course of the day, they get sicker and sicker. That evening, kids come home from the playground also feeling sick. The next morning, kids go to school, and everyone's sick. Parents had no idea that sending their child to school could be unsafe. Cases pop up across the country and across the world. The world's launched into utter pandemonium as parents are forced to stay home with their kids, leaving grocery store shelves unstocked and leaving the healthcare world undersupplied. Still, kids get sicker and sicker. This sounds drastic, but we lived through it. The COVID-19 pandemic saw 6.05 million deaths, not including the 1.4 million indirect childcare deaths due to food supply issues, due to healthcare shortages, and so on. There is a 64% chance of a pandemic as severe, if not more severe, than COVID the next 50 years. That's a statistic that Duke Global Health published, but it's actually an underestimate because it assumes the probability that the pandemic occurs stays the same over time, but it's not. As the world becomes increasingly more connected with trade and globalization and population growth, we're actually going to see an increase in pandemics like we have in the past. What does this mean? It means that 64% is an actually gross underestimation of how risky a pandemic is, meaning that accurate diagnostics are even more important. Existing diagnostics should be rapid, affordable, accessible, and accurate, but they're not. They are slow, they're expensive, they require a centralized lab network, but they are accurate. Our device addresses the needs we've identified in the healthcare system. MEViD takes 10 minutes to run from start to finish, is very cheap, costing $1.50 per test, can be deployed worldwide because it does not require a centralized lab network, and has parallelized tests, meaning it can run 6 tests at once.
Thank you, Carisa. Now I'm going to briefly take you through how our device works. So first, the patient spits into a small tube, which then slides into our compact device to be further analyzed by our hardware and software detection algorithms. The results are then displayed via Bluetooth to be displayed on our app interface for viewing by either the patient or the test administrator. All of this happens within 10 minutes at a fraction of the cost of traditional diagnostic technologies. So in designing MEViD, we wanted to create something that could be widely deployed across the world, and particularly to resource-constrained settings. Something that could operate within a wide variety of both clinical and non-clinical settings in order to really streamline these mass testing efforts. So some of these settings that we really view MEViD as being useful in include airports, schools, community centers, and clinics. The other piece of accessibility that we really tried to optimize for here was a multi-channel capability, or the ability to conduct multiple tests at the same time. This ensures that MEViD can be rapidly adapted to the changing needs of a given situation. So for instance, in the case of an unknown viral threat, we might choose to test one patient for 6 different tests, and as we narrow in on what that viral threat is, we might choose to test 6 patients all for the same virus. So we defined some needs specifications around 5 key components of accessibility that we identified, which you can see here. Distributability, or how small our device is, how intrusive it is, usability by non-experts, adaptability with existing electronic medical record systems, and lastly, modularity, or in other words, can we swap out these biosensors to test for the different viruses that we need to test for? All of this will ensure that MEViD is accessible enough to be widely deployed across both the clinical and non-clinical settings that I mentioned earlier. And so now I'll pass it off to George to talk more about the underlying tech.
So we sort of walked through how we think this is going to be a great solution for pandemics in the future, and you might be curious, how exactly are we doing this? What is the underlying technology? And that's what I'm going to take us through a little bit right here. So MEViD is designed to work with an electrochemical biosensor designed by the De La Fuente lab at Penn. The sensor consists of viral receptors parked on a graphite electrode, which are able to sense viral binding and change the behavior or current flowing through the electrode. When there's no virus present, current is able to flow easily through, and you see a higher peak on our output graph. However, when virus is bound, current flow is impeded, and you end up seeing this lower red line on our graph. This is the basis by which viral diagnostic is possible with MEViD. MEViD employs potentiostats, which are analytical instruments that sends varying levels of voltage through the electrode and reads out the corresponding current. These are typically benchtop large-scale devices. To achieve the multi-channel testing of 6 tests at the same time, you will need multiples of these devices plus the support of desktop computing. But we've managed to miniaturize our design suited for a particular purpose, optimized to work with the pen biosensor into one small device that actually is smaller than the size of a typical potentiostat. The De La Fuente lab sensor was originally developed for the COVID-19 pandemic, and it uses COVID's ACE2 receptor as its underlying technology. However, it is important to highlight that this is a modular platform, and any viral receptor can be used in place of COVID's ACE2 receptor. This is an important characteristic for us as it enables a wider range of testing capabilities across viral threats, making multi-channel functionality a truly valuable aspect of our MEViD device.
Great. And now we're going to talk a little bit about the results of our system. So in order to ensure that we could be as accurate as needed with our small-scale potentiostat, we replicated an experiment that was done by a De La Fuente lab in order for them to determine their limit of detection. So we first start off with a blank sample and see this parabola, and then as we added increasing concentrations of spike protein, we're able to see the expected decreased signal height, and the correlation between the height and the log concentration of spike protein was really tight with an R9 of 0.99, which is very similar to what the De La Fuente lab had when they ran their clinical trial, which resulted in 100% accuracy with saliva samples relative to PCR.
Just to walk through some of our design process, now you've seen the whole scope of our project. We started with the proof of concept. This was really just a single potentiostat system and a very basic software application. We then expanded this to have the integration between our hardware and software and upgraded this to a dual potentiostat system for our minimum viable product. Our final prototype now includes a 6 potentiostat system, so the ability to test 6 things at once, a full detection algorithm on the software side, and electronic medical record integration. We used Epic Sandbox as a proof of concept there. And then as we go to next steps, commercialization, we see this on a 3-pronged business plan. So when we think about how we want to commercialize, we could see this as an over-the-counter product, we could see it as a primary care product, or also as a proof of concept, point of care product. So in the first case, we would use the local storage aspect of our product, and this could be sold over the counter at like pharmaceutical outlets. In the second case, we would use cloud-based storage and also this EMR integration for back office functionality and HIPAA compliance. You can think of a standalone physician office or a clinical system such as Penn Medicine. And then in the third case, we have point of care. This would also use cloud-based storage. And because of the cheapness and also the rapidity of our test, this could be really just reach a wide amount of people globally. Also could help combat future pandemics for other government organizations or health organizations. MEViD could be deployed commercially in all 3 of these aspects. Thank you. We are MEViD, together fortifying our disease response 6 tests at a time.
So it doesn't surprise anyone to say that this is an area of enormous interest right now, and there's just billions and billions of dollars going into figuring out how to do this quickly. And it seems, there are a lot of competing products out there. One aspect that I think would be great if you can go into it a bit more would be how long does it take to take this platform technology, as you mentioned, it has sort of the modular functions, and create a new sensor, a sensor for a new virus that doesn't exist yet. And if you can't speak to it, that's fine. But manufacturing-wise, how long would that create?
You want to talk about the other viruses?
Yeah, so it's already, the same technology has been used by other labs to detect MERS, H1N1, avian flu, Ebola, like a long list of diseases. The lab itself, it took about a month for them to replicate the same thing with herpes. That was again done with 2 PhD students. So you can imagine if you're really commercializing it, you could speed up that process more than what 2 PhD students can do.
Yeah, so essentially this is not the first time that we faced a threat like this in some lab somewhere in the world came up with this idea to use electrochemical testing, but it's never been progressed to a place where you have the device to be able to take it to market and have this multi-channel functionality. There is going to be challenges on the manufacturing side in terms of manufacturing the sensors at larger scale. They use very cheap materials, graphite basically out of a pencil, but the machining and the processes required needs to be created on a more commercializable scale. So that's probably the one that's going to take the most time.
I'm probably going to piggyback off of the question that was previously mentioned. I know this is based on the Fuente lab. Are there other tests you're looking at that are non-saliva based? For example, direct tissue swabbing as well as phlegm samples?
Yeah, so they've replicated with nasopharyngeal samples. That had a little bit lower accuracy, but what type of sample you want to use, it doesn't matter that much in terms of our device. It's more wherever the virus particle is in highest concentration.
I'm going to ask less technical. Why 6? What's the trade-off?
Honestly, the reason we first did that was because at that point the lab was exploring 6 different viruses. But it was more just as a proof of concept. The fact that we can multi-channel it at like a high number was basically the whole idea, and we didn't feel like soldering enough for 100.
Soldering 6 is a lot.
Yeah.
Yeah, and our device, I guess it's important to mention that the device is designed 2 channels at a time, so it's very easy to just print more of the PCBs and add, say, 10 channels into the same device. You just have to change the casing.
Hi. So you mentioned in your future plans that this could possibly be distributed at like a Walgreens, right? So you're talking about, non-medical folks using this device. What, where does it stand today versus where would you need to get it to be patient-friendly and being used by an everyday, person?
Yeah, so in terms of what we're thinking for Walgreens, we'd probably start with more even the point of care aspect instead of the over-the-counter shelves, but honestly they have minute clinics and that's been a huge emphasis there. So they would have one of our devices and then you could sell tests and then so patients would come in and then get the test there. So in that standpoint, really the usability of our app, you can teach it to someone in under 5 minutes. So the idea there is once you have the device and the receptors in, it's pretty easy to integrate between those. So that would be the idea there. To get it to like an over-the-counter product where any family could walk into the store and buy the whole thing, we'd probably want to, 1, miniaturize it even more. 2, we'd probably want to come up with some testing protocol where you buy tests in addition to the actual casing of it, almost like packs, and then you could change them out or buy different tests as you see fit. So that would take some more thought as well.
Hi, I had a question about the adaptability of this device with mutations, especially considering COVID and the different changes to the spike protein. So I'm wondering, this has a very advanced coupling reaction, I suppose, of the receptor to the gold nanoparticles. So I was wondering if you could speak to that. Also, what about the storage conditions for this kind of device? You're having proteins, antibodies, at least right now it seems like it's only receptors. How do you keep them stable enough to make good measurements?
Yeah, so for the, with changing mutations, the really convenient thing is that as the viruses evolve, the binding between the virus and the receptor tends to be tighter, which makes our device work better. So if you look at the data for this device with like alpha variant versus beta variant, much easier to see beta variant, much easier to see delta variant. And when we were first looking at the experiment, we were thinking about actually being able to differentiate between the two. We didn't end up getting different spike protein for all those viruses, so we didn't do that data, but it would only get easier over the course of evolution, basically. And then for storage, you need to store it at 5C, which, yeah, just like a refrigerator. There are stability issues, like you can't store it forever. And if we were to commercialize it, we'd want to look into more preservatives and how to keep it longer.
The other important thing to mention there, can you hear me?
Yes, I can.
Great.
And sorry, I'm listening and checking something.
No, just want to make sure the mask doesn't limit projection. The other thing is that a lot of tests right now that exist, if there was a new COVID beta comes out, right, you'd have to completely change diagnostics, including the actual physical manufacturing itself. What ours is unique with is that you don't have to change a casing, you don't have to change any of the electronics. It's a software update to the phone. And that's all that changes, besides the biosensor, and that's the production capability. But that theoretically would make it easier to distribute, and given that our device will hopefully work in places that are more delocalized and don't have access to pen level resources, the level of dislocation required to actually run the device is really important in ensuring functionality on a broader scale.